please ask questions in the chat section. And of course, if you want to ask questions live, then after the talk, please turn on your camera and your microphone, and you're more than welcome to ask questions to our speakers. I ask all the speakers to please take in consideration the time that you have. Try to, well, not try, no, please keep to your time, and if need be, I'll let you know if you're going over time. So we'll start with our first presentation from Mohit Kumar, who's assistant professor at the Indian Institute of Sciences in India. And the title of his talk is Stemness in the Hybrid Epithelial Mesenchymal Phenotype, a hallmark of high metastatic potential. So thank you very much, Mohit, for accepting the invitation, and we're very much looking forward to your talk. So the floor is yours.
Thank you very much. And I would like to thank IRA and the organizers for the opportunity to present some of our work that we have been doing. EMT, as mentioned, is not new to this audience. The only reason I present this slide will become clear in a minute. These are snapshots taken from various reviews that have been written about EMT, the previous decade. And what you see here is that most of these reviews, which reflect the schema of the thought process in the field, treat EMT as a binary process. So either cells are reported as completely epithelial type junctions and adherence junctions and not really moving, or they are usually reported as a single cell mesenchymal movement. There were some ideas about this hybrid state in between, but it was discarded as being an intermediate and not a final state in itself that cells can take on during their entire metastatic journey. So this is the question that we started working on, with asking whether EMT or MET is really just a binary process, or can there be more states in it? So at that point of time, what we did was looked at different molecules, transcription factors and microRNAs, which are known to regulate either EMT or MET across different carcinoma contexts, and then put together into a network and formulated a mathematical model to reflect the interactions that you see here on the screen. So here, as many of you may know, there are these two transcription factors that drive a more mesenchymal phenotype, and these two microRNAs that drive a more epithelial phenotype. And what you see between them is a tug of war, or a toggle switch, or double negative feedback loop, which is abundantly seen in various developmental decision-making processes when a progenitor cell has to decide which cell to be its final fate. The numbers here represent some quantitative input-output relationship taken from experimental data as available at that point of time. So then we constructed this mathematical model where the general framework is that one or more microRNAs can reversibly bind to a given mRNA molecule. And once this mRNA microRNA complex is formed, then it can either just lead to translational inhibition for some period of time, and then the mRNA is again available for translation, or it can actually degrade the complex itself, because microRNAs can do both things. So long story short, what were the predictions of our mathematical model? It's shown in this diagram, where the x-axis can be thought of as some EMT-inducing signal, and the y-axis can be thought of as the measure of EMT-ness. The solid lines show stable states. The dotted curves show unstable states. We'll only worry about the stable states, because that is what would give rise to a phenotype. So what you see here is clearly these two phenotypes, which were experimentally well known, very low levels of ZEB epithelial, very high levels of ZEB mesenchymal. But we also found that there was a third state which was possible in between, which had intermediate levels of ZEB as compared to epithelial and mesenchymal. We chose to call this the hybrid epithelial mesenchymal phenotype at that point of time. And the idea was that this particular state, this stable phenotype that cells can potentially achieve, is likely to have combination of epithelial and mesenchymal properties. So therefore, it is likely to migrate collectively instead of single cells, which is typically a process of a more mesenchymal state. So that's about the graph looking horizontally. I'll draw your attention to look at the graph vertically now. So here in this region, if you draw a vertical straight line, you cross only one solid blue line, which corresponds to epithelial state. On the rightmost part of the graph, again, if you draw a vertical straight line here, you again cross only one solid blue line, which corresponds to the mesenchymal state. But in between, you see regions in which, if you draw a vertical straight line, you would cross more than one such curves. What does that mean? is that depending on where you are in this diagram, it is possible that cells with the exact same genetic background can have different phenotypes. Of course, there can be completely epithelial cell lines and completely mesenchymal cell lines, but there can be cell lines in which you actually see all the three phenotypes present, and also they can switch back and forth among one another. So those were the three major predictions: that A, EMT is not binary. You can have cells in a hybrid state, stably maintained, and then you can have this non-genetic heterogeneity and also switching between those. So we worked with a couple of collaborators. This is with Sam Hanach's group at MD Anderson Cancer Center, an expert in lung cancer. So this is a non-small cell lung cancer cell line stained for E-cadherin and vimentin, the canonical epithelial and mesenchymal markers using red and green respectively. So what you see in this image is that most cells actually do have both red and green, suggesting that it's not just a mix of epithelial and mesenchymal cells, but at a single cell level, these cells are reflecting a hybrid phenotype. And they continue to do so over multiple passages in vitro. So it's actually a stable system, not necessarily just a snapshot. Flow cytometry using the same two molecules done for various different cell lines belonging to different cancers also revealed the same thing, that yes, despite having the same genetic background, you can have this heterogeneity. And interestingly, this particular experiment, which was done using prostate cancer cells, where these three cell types were segregated, the mesenchymal one, the epithelial one, and what they call EMT, but it's actually a double positive, so a hybrid phenotype. And when they were cultured independently, what was observed over a period of 2 weeks was that each of them was able to give rise to another. Of course, the frequencies were different, but they had this possibility. So without any induction, you can still see cells that can switch back and forth. And those were all the three predictions of our model, which have been validated over the past few years. So what has happened now, again, long story short, is that people have started thinking of EMT as not a binary process, but as a process where you can have many states in between. This is work done by Cedric Van Payne's lab, represented here. They looked at squamous cell carcinoma, but in other cancers also, these states have been identified and the molecular characterization is currently ongoing. But clearly we have come a far way from being a binary process to one with there are many states that are there in between. And interestingly, which is what I'll focus upon, is that these hybrid states that have been identified since are actually shown to be the most stem-like or the most metastatic as compared to the ones which are completely epithelial or completely mesenchymal. So once we started modeling EMT, there were various other groups that started looking into this question, mathematicians, physicists, computer scientists, chemists, etc., each coming with a slightly different perspective, using a slightly different network of varying sizes, different modeling strategies, etc. But the common theme was still the same, that even if you look at a larger network, because clearly what I presented to you earlier is a very small network of EMT, and EMT is clearly much more complicated than that. But even if you look at a larger network, this general picture is still reserved. It's still that feature is conserved, that you have these predominant epithelial and mesenchymal states, but you can have one or more hybrid states in this network. Now the question was, how does this affect stemness? So at that point of time, when we started looking into this 6 to 7 years ago, there was this hallmark paper from Bob Weinberg's lab suggesting that EMT increases stemness, which is shown in this diagrammatic form here. EMT as the axis, left part being epithelial, right part being mesenchymal, and the stemness window lies somewhere to the right. There were papers which said the exact opposite thing, that EMT decreases stemness, and then a paper by Max Feature's group saying that, well, EMT may not have any correlation with stemness because you have both epithelial and mesenchymal stem cells. So this was very puzzling to us. Again, we did a similar aspect. We took what were known to be important players in the context of regulation of stemness, see how they connect to EMT, and made a mathematical model. What our model predicted was that actually the stemness window is most likely to lie somewhere in between. And the position of this window is not a fixed entity. It can move left or right, but it's most likely position where it spends maximum amount of time is somewhere in between. So how does this explain all these potentially confounding and confusing and contradictory data? Is that all these papers are still from a time zone where people were thinking of EMT as a binary process. So you really needed to acknowledge the presence of hybrid cells and segregate them and not let them be miscategorized as either epithelial or mesenchymal. So when people started doing that and identified hybrid cells, clearly saw both in vitro as well as in vivo that these hybrid cells were the ones which were actually forming maximum tumors. And this theme has also been shown by various other groups more recently. In fact, the latest paper by Bob Weinberg's lab claimed that hybrid cells are essential to form tumors. They started with, they did experiments where they co-cultured epithelial and mesenchymal cells together, but still those cells were not able to form as many tumors as a small percentage of hybrid cells. And this is an in vivo experiment. So if we go ahead with this hypothesis or this framework that yes, the hybrid cells are the most plastic, they are somewhere in between. So of course, they have the maximum degree of freedom to switch to either epithelial or mesenchymal. They have higher stemness. And there are other reports which show that they may be equally, if not more, resistant to drugs and matrix deprivation as that of a mesenchymal state. So the question we started asking here was that, is there something which is very sacred in terms of the connection between hybrid EM and stemness? If you just look at the network topology itself, does something emerge out of it, irrespective of the parameters, the kinetic parameters, such as, the rate of production of a particular protein, the rate of degradation of a particular protein. So, of course, in every cell, these rates of biochemical reactions are going to be different. So if we take a large ensemble of these values and look at what shows up, is this theme that hybrid is more likely to be associated with stemness? Does it show up there? Or is it just an artifact of some specific parameters that we had considered earlier? So this work was done by two students, a medical student and an undergraduate student at ISC. And we looked at the circuit that I showed you earlier, the microRNA 200 and ZEB representing the EMT axis, and LIN28 LET7 representing the stemness axis. So we simulated this now using a much larger number of parameters. So we see more states. Earlier we saw only 3 states. Now we see 2 hybrid states, and these are what are shown in this heat map here, where the levels of all of these molecules are plotted. And what you see is that clearly there is this epithelial state with high levels of MER200 and LET7 and low levels of ZEB and LIN28. And on the other extreme, you have the exact opposite state, which has low levels of MER200 and LET7 and high levels of ZEB and LIN28. And in between, you see there are 2 other states. We call them hybrid epithelial and hybrid mesenchymal, depending on the distance they were away from epithelial or mesenchymal states. Then we calculated the probability that each of these states is associated with stemness. So what you see here in this diagram is that among these 4 states, the hybrid epithelial and hybrid mesenchymal states were more likely to be associated with stemness as compared to epithelial and mesenchymal. We are not ruling out the possibility that the 2 extremes cannot be stem-like, but these states in between are clearly more likely to be stem-like is what we are saying. And this is something which is embedded in the topology itself. Now, one other thing which we looked at earlier was trying to identify factors that can actually stabilize a hybrid state. So this is the same diagram that I showed you a couple of slides ago. What you see here is that there is a region in which the only phenotype is epithelial. There is a region in which the only phenotype is mesenchymal, but there is no region in which the only phenotype that is available to cells is the hybrid phenotype. So hybrid is always coexisting with others. So then what we looked at is in developmental biology context, are there players which are known to stabilize hybrid cells? Are there players known to stabilize a collective cell migration? So here we added one such player, and we saw that the bifurcation diagram that I showed you earlier changes. And now you have a region in which the only phenotype that is available to cells is hybrid, which is shown by this dotted line here. So suggesting that if this molecule is present in cells, then hybrid is likely to be more stable. And that's why we call this a phenotypic stability factor. It's not an inducing factor because the hybrid cell was present earlier also. We identified various other such factors for which we have experimental evidence that supports it. I'm going to show you one example of that, where we again went back to the same cells that I showed you earlier, the H1975 lung cancer cells, and we deleted one of such stability factors, and we see that the EEC adherent levels go down drastically and cells have switched on to a completely mesenchymal phenotype, which is also evident in their migration pattern, that the collective migration gets disrupted and these cells are moving as single cells. So these factors are behaving as a brake on EMT, and as soon as you remove that brake, you see that those cells undergo a complete transition. Now, clearly, we know that these particular stability factors, if you overexpress them, at least GRHL2 and OPIL2, can drive a mesenchymal to epithelial, the exact opposite transition. So we looked at the core circuit that I showed you earlier and added these different molecules based on the topology that we know about them. And what we saw was that, yes, if you overexpress any of them, which is the last column corresponding to this, you clearly see a decrease in mesenchymal, an increase in epithelial, and in some cases you also see the hybrid epithelial and the hybrid mesenchymal populations increase. But what did not change was the association of hybrid EM with stemness. So irrespective of which circuit you look at, the control case that I showed you earlier or the one where we have incorporated these components, the probability that a hybrid state is stem-like is always greater than the probability that a non-hybrid state is stem-like. And here, when I say hybrid, this is hybrid epithelial and hybrid mesenchymal pooled together, and non-hybrid is epithelial and mesenchymal pooled together. So these molecules which are capable of stabilizing the hybrid EM state, these molecules, when overexpressed, can push the cells to a completely epithelial state. They do have this ability that they maintain the association of hybrid EM with stemness. But as I said earlier, that the connection of hybrid EM cells with stemness is not exclusive. We are not saying that every single cancer stem cell has to be a hybrid EM, and we're not saying that every single hybrid cell has to be a cancer stem cell. All we are saying is that the likelihood, the overlap of these two features is somehow the hybrid on the EMT axis and cancer stemness here. They are likely to much more associate with one another as compared to anything else. I'll just summarize here, used mathematical models as a tool to help decode the intracellular dynamics of regulatory networks that control the epithelial mesenchymal plasticity, or which is what EMT is beginning to be called much more frequently now, with the ideas that there are many more states than just two. And we have predicted this, and in vitro, in vivo evidence has clearly validated the predictions of our model that the hybrid EM cells are more stem-like, and the association of this is not exclusive. And there are recent clinical reports which actually show that even if we have a very small percentage of hybrid cells, that is apparently sufficient to give rise to a worse prognosis of patients. With that, I would like to thank you all for the attention, and I would like to thank SERB for funding.
Thank you very much, Mohit. I ask if anyone has their microphone open to please close, because there's some noise in the background. So first of all, a wonderful talk. In fact, I was very excited for your talk today, because it's something that we've been looking at in the laboratory also, and this whole EM epithelial mesenchymal hybrid state, I think is quite interesting when we're talking about STEM. Please, anyone has any questions, please, you can either open up your microphone and camera and ask Mohit directly, or you can use the chat. Until we get our first question, I actually would like to ask you a question, if you don't mind. So a lot of times these studies are performed with in vitro. There's a lot of noise in the background. Can you hear me, Mohit?
Yes, I can hear you.
Okay. My question is, a lot of times we do these cells, these sort of experiments with in vitro cultured cells, and we completely disregard what's happening in the microenvironment, and we know the microenvironment plays a very important role. CAFs and macrophages secreting factors in inducing EMT state, and also promoting or modulating stemness. So how do you take into consideration the influences that the tumor microenvironment may have in this hybrid state, whether or not it's also recapitulated in vivo, or whether or not you see it might be more in vitro phenotype, or likely in both cases.
That's a very important question. Thanks. So one thing that we are developing models now is to try to quantify how different percentages and durations of hypoxia might be affecting EMT. So, because again, when you talk of hypoxia, we know that sure, HIF1 alpha gets activated and then it activates a whole bunch of genes downstream. But is there a difference in how cells adapt to hypoxia being there for, say, 2 hours versus 2 days versus 2 months? So what we see so far is that if hypoxia is in a more oscillatory manner, then the hybrid state seems to get more stabilized as compared to when it's there for a continuous manner. The other feature that we have looked at is cell-cell communication within the tumor cells themselves. And we have identified that notch-jagged signaling instead of notch-delta signaling actually helps stabilize hybrid cells at a cellular, sort of at a community level. And jagged was actually found to be among the top 5 genes which were upregulated in cells which were moving collectively. So if you look at CTCs in vivo, then that are moving collectively versus the ones that are moving individually, jagged was one of the top 5 ones. And if you downregulate jagged, then what we saw in our case in inflammatory breast cancer cells was in this in vitro experiment was that there was no difference in proliferation or death, but there was a marked difference in tumor formation. So we are able to somehow destabilize hybrid cells if we sort of cut down the notch jagged but not the notch delta aspects. Those are 2 things we have looked at currently, and we are in the process of incorporating macrophages M1 versus M2 to begin with in our models to start to see how can we develop these in silico co-cultured models and make further predictions.
Thank you. It's extremely complex. We have a number of questions that have come in the chat. We'll try to answer as many as possible in the time. The first one from Teresa. She says, thank you for that wonderful presentation. Did you look at invasive properties of these hybrid cells? So for example, if they secrete higher protease levels like cancer stem cells?
No, we have not looked at that.
Okay. Another question from Joanna. Thank you, Mohit, for your nice presentation. What do you think that stabilizes the hybrid phenotype in some cancer models? Genetic alterations or secreted factors from the tumor cells or from the microenvironment?
I think it's a mix of all of them, and we are trying to quantify in different models what is the relative contribution coming from each of these. So far we have identified NRF2 molecule or pathway as the only marker which is actually maximum in hybrid. So far, all the others, the GRHL2, OVL2, ZEP1, are either maximum in epithelial or mesenchymal, and they follow sort of a monotonic trend. But NRF2 is low in epithelial, low in mesenchymal, and peaks in hybrid. So we're still trying to identify, and NRF2 clearly known, given its role in metabolic reprogramming, we're still trying to identify, can there be a metabolic signature of these hybrid cells?
Yeah. And another question from Farah. She says, this person says, thank you, Mohit, great talk. How you predict the aggressiveness of the EM hybrid cells?
So in our case, we considered the levels of OCT4 as a measure of aggressiveness. And based on embryonic stem cell literature, we know that if OCT4 levels are too high or too low, they usually tend to differentiate into different images. So that was how we constructed the Stemness window.
Okay. And Adit also wanted to ask a question. I don't know if you want to ask the question live, Adit.
I would prefer so instead of writing.
Sure, go ahead.
It's easier. Thank you very much. I wanted to ask you about the variability of EMT states, because it might be that cells have one phenotype of EMT and another cell has another phenotype of EMT, and not necessarily both, even in the cell tumor type, not necessarily both markers of EMT are expressed together. So how do you consider this variability in EMT phenotypes when you connect it to stemness?
That's an excellent question. So far, we have considered E-cadherin and vimentin, but yes, as you rightly said, that there can be different markers and which can change. So some cells might be, say, higher in vimentin, but not in fibronectin, or vice versa. So that is something we still need to carefully do. So far, we have done it in a sort of very low throughput manner. We have not gone into that high throughput analysis. And in that case, we have used the two most canonically usually agreed upon markers, E-cadherin and vimentin.
Okay, thank you.
Thanks.
Fantastic. You have a few more questions in the chat, and I encourage if anyone, if they want to continue asking questions, you can chat privately with Mohit. So Mohit, if you don't mind answering some of those questions in the chat,
Definitely, sure.
that will allow us to move on. But excellent talk. You obviously have created a lot of discussion and a lot of questions. So I think you'll have